Polypeptide synthesis. Polypeptide synthesis is the biological production of peptides, which are organic compounds in which multiple amino acids are linked via peptide bonds. They are the building blocks of proteins which are essential for cell function. Polypeptide synthesis is important for creating proteins that carry out different functions in our bodies. DNA, deoxyribonucleic acid, is made up of a double helix structure, which includes a sugar-phosphate backbone and nucleotide bases, adenine, thymine, cytosine, and guanine. These bases are complementary to each other, with adenine always pairing with thymine and guanine always pairing with cytosine. These bases are bonded together by hydrogen bonds. The process of polypeptide synthesis involves two steps: transcription and translation. Transcription occurs in the nucleus. This process involves the information of a strand of DNA being copied onto a molecule of messenger RNA, mRNA. The process of transcription occurs in three steps: initiation, elongation, and termination. The first step of transcription is initiation, which is the process of the DNA strand unzipping. The section of DNA that codes for the particular protein will unzip after the RNA polymerase slides across the DNA strand, breaking the hydrogen bonds between the bases. This results in two strands, one being the coding strand and the other being the non-coding strand. The next step of transcription is elongation. This process involves the RNA polymerase, which slides across the template strand, attaching free RNA nucleotides to the non-coding strand. Guanine and cytosine attach to each other, and adenine attaches to thymine. However, in the case of RNA, thymine is replaced by uracil, which now attaches to adenine. The last process of transcription is termination. This involves the nucleotides reaching a stop code in the gene, which here is UAA, and the newly made messenger RNA detaches from the strand and leaves the nucleus, entering the cytoplasm for the next step, translation. Once the messenger RNA enters the cytoplasm, the process of translation now occurs. Translation is the process of translating the code of the messenger RNA to the sequence of amino acids to form a peptide bond. The first step of translation is initiation, where ribosomes bind to the messenger RNA strand to produce a complementary transport RNA, tRNA, anticodons. These anticodons each have a specific amino acid which attach to the codons on the mRNA strand. Elongation is the second step of translation. During this step, the ribosome moves along the strand and tRNA anticodons and the amino acids are attached to a growing chain of acids, which are called peptide bonds. These peptide bonds grow to form a polypeptide chain. The final step of translation is termination. Once the tRNA reaches the stop codon, the polypeptide chain is released into the cytoplasm. Here it either folds to form a protein or bonds with another polypeptide chain to form. Through correct polypeptide synthesis, this ensures that the correct proteins are made. However, when there is a mutation in the sequence, this creates a change in the amino acid sequence and can totally change the function of the protein being made or stop that protein from even being made. A mutation is a change in the DNA sequence. A point mutation is a type of mutation that only affects a base pair only. A missense point mutation is when the base is altered or changed, resulting in a change in codon, coding for a different amino acid. Here the left side is showing the original sequence compared to the mutated sequence on the right. Here the right nucleotide base on the DNA non-coding strand of guanine is substituted for cytosine on the mutated side. This results in the wrong base pair on the mRNA strand, resulting in the wrong anticodon coding for the wrong amino acid. Depending on the protein made from the wrong sequence of amino acids, it can change the continuity of the species. It could change the protein for better, impacting the individual, allowing them to adapt to the environment and live to reproduce, or impact the individual badly, resulting in a disease or disability affecting their chance of survival onto reproduction. If a mutation occurred in the somatic cells, it will only impact the individual and not be passed on to the offspring, where if the mutation was in the gamete cells, it will be passed on to the offspring. Sickle cell anemia is a type of disease caused from a missense mutation, and is when the red blood cells are shaped abnormally, resulting in restricted blood flow, has an average lifespan of 42 to 47 years, and has to be inherited from both parents for the offspring to contain the gene.